You know who it is. Professor Jay here. Hey, today, guys, we're going to be talking through a specific protein synthesis example. Now, a lot of you have learned protein synthesis before, right? It's the process of getting DNA, the instructions for your body, onto mRNA, so the messenger RNA, which will go to a ribosome and it will make a protein and all. What does that have to do with anything, right? So we're always confused. I was confused when I was in school. But what's the point? What's the purpose of that process in general? It's called the central dogma, the central idea of biology. So, where does it fit in? Let me show you. So, I want you to focus in right here first, okay? So, I drew kind of a cross section of your lumen. Well, I guess this is kind of a frontal section of your lumen and your intestines. So, you've got a bunch of cells lining the lumen. So, that means basically hole or opening in your intestines. So in this lumen, there will be nutrients that you ate, that you digested, and it will come in and fit right next to all these cells in the intestines. And the job of the intestinal cells, the job of each and every one of these is to absorb, to bring in the nutrients like glucose and sodium. So you eat this food, comes to your cells, the cells need to bring it into their cell bodies and then distribute it throughout your body. Okay, Professor J, what does this have to do with protein synthesis? Let me show you. Okay, so I'm going to step aside. I want you to get kind of oriented here. So I'm going to zoom in on one intestinal cell. So right here, we've got an intestinal cell, this big box with kind of the projections here. And I've got the nucleus, I've got some rough endoplasmic reticulum, and obviously there's going to be a lot more things in this cell. But these are the main three players. So you've got your nucleus, rough ER, and then you've got your cell membrane, that phospholipid bilayer, right? And you have all of this glucose, so all of these nutrients and the sodium, and again, there will be more nutrients, but this is just an example, floating outside in the lumen. What was the goal again? We need to bring these into the cell. Well, right now, these guys, first off, sodium is a charged ion, so it can't pass through very easily. Glucose is a pretty big molecule, so it can't just pass through the phospholipid bilayer of the membrane. So what are we going to do? I'm glad you asked. Well, first off, we need to find a sort of garage door to bring these guys into this cell. What do you think their garage door is going to be? A protein, right? So in order to make a protein, we got to start with the instruction manual, the DNA. So we're going to go in the nucleus and we are going to find a gene for a protein that is a glucose sodium symport protein. So what this protein does is it's going to, sim means kind of the same, so it's going to transport in the same direction both glucose and sodium into the cell. Well, how do we get that protein? I'm glad you asked again. Well, when you go into your DNA, you're going to have your gene, which is your short DNA segment that codes for a specific protein. mRNA polymerase, the enzyme that builds mRNA from that template, is going to make an mRNA molecule. And this is basically the recipe, okay? So the recipe of that mRNA, where'd my marker go? There it is. That recipe of mRNA is going to leave the nucleus. So here you got this kind of long winding mRNA molecule. And again, this is the recipe for that symport protein. Well, it's not good enough to just have a recipe, right? You got to do something with it. So you take that mRNA and you run it through one of these dots. Those black dots are the kind of dotted things inside of the endoplasmic reticulum, is called a ribosome. So all of these guys are ribosomes. And these are the little chefs of your cells. So they are going to take the recipe, the mRNA, and they are going to chain amino acids together, the base unit of proteins, to develop a full functioning protein. And let's say that the protein for this symport looks something like this. So it's kind of like a barrel shape. And maybe that's what the protein looks like, kind of tiny. Well, it's no good just sitting there, right? So what has to happen, and this is kind of a complex process, is a vesicle, it's basically like the cell's bus system, is going to form around that. It's going to be a phospholipid bilayer, and it's going to form around that protein. Well, where do we need it to go? Do we want to go to this side? or this side? Well, probably this side because this is where all the nutrients are, right? So what's going to happen is this vesicle is going to fuse with the cell membrane on the outside. And once it fuses, it will look something like this. So now we've got this protein embedded into the phospholipid bilayer, and now it can get to work. So the goal, once again, of this protein is to symport, bring in glucose and sodium. So this one specifically, I forget what the name is going to bring 2 sodium and 1 glucose per round. And it does this rapidly, super fast. So now all of a sudden we've got all this sodium, bring that in here, and glucose, these are not drawn to scale, into the cell. Because we made that protein to get into the cell membrane to bring those molecules in. And eventually, what will happen is these guys will be transported to the basement membrane of the cell to eventually go into the capillary bed, which is your blood vessels, and then go all the way throughout your body to nourish the rest of your body's cells. So, once again, this was an example of how protein synthesis, making a protein from DNA into mRNA, into a ribosome, chaining amino acids together, making a protein can function to help your body as a whole. And there are thousands of examples of these proteins helping your individual cells, as well as facilitating other processes that your entire body needs in order to maintain homeostasis and keep you alive. Well, I hope this was helpful. Drop any questions you have in the comments below, and we'll talk to you soon.